that stuff has been cell physiology. We're finally ready to start our first system, the nervous system. All right, so we're going to start our first system, the nervous system. In order to understand the nervous system, we've got to go back and review the structure of a neuron. When we look at neurons, we find that these are special cells that can conduct electrical impulses. Right? They're specialized cells that can conduct electrical impulses, which is pretty different. It turns out muscle cells can do this as well, but being able to conduct electrical impulses is really special. Also, when we look at these neurons, they have some modifications that we don't typically see in our typical cell. So, here we go. Here is a typical neuron, if you will. When we look at that neuron, we find that there is a nerve cell body, and that nerve cell body we're going to call the perikaryon. Karyon means nucleus. Peri means around. So this is the part that surrounds the nucleus, the perikaryon. The extension that comes away from the nerve cell body is something called an axon. And then there may be small extensions that lead to the perikaryon, and we're going to call those dendrites. They bring nerve impulses in. Inside the perikaryon, we're going to find things that we would expect to find. A nucleus, ribosomes, mitochondria, all those kinds of things. Right? They're all in there.
Which one's the perikaryon again?
Perikaryon is the nerve cell body.
Okay,
So that's this that's around the nucleus. Around karyon nucleus. The arrow's trying to show you that the dendrites bring impulses to the nerve cell body, and this arrow is trying to show that the axon carries impulses away from the cell body. The area that leads to the axon is a special area that we call the axon hillock. If you look at your diagram, it's there. It's hard to see, but it's there. We also might see coming off there may be a collateral axon. So once we get out here to the axon, there may be branches. Here's another collateral axon coming off. At the end of the axon, there are lots of little branches called telodendria. Right? Lots of little branches called telodendria. And at the end of the telodendria are synaptic knobs. Synaptic knobs. Synaptic knobs are going to have neurotransmitters in them. We're going to spend a bunch of time next week talking about that. I always thought it'd be a great name for a girl child, Odendria. Right? Don't you think? It's a pretty name. Sounds nice, Odendria. And right, if any of you are pregnant and have a kid, if you name your daughter Odendria, I'll give you an extra 10 points. When people ask you, why did you name your daughter Odendria? You say, well, I was taking physiology. It sounded pretty, right? Odendria. The real thing that would really be nice with that, right? If somebody calls and Odendria is not there, and you just say, well, tell Odendria I called. All right. These are the branches. All right. Around the axon, there is a modification that we call the myelin sheath. Right? So wrapped around the axon is this thing called the myelin sheath. The myelin sheath, at least in the peripheral nervous system, is produced by specialized cells that we call Schwann cells. Right? So it's produced by specialized cells called Schwann cells. Well, let's take a look at this. So here's another diagram trying to show there's the axon, but here's this myelin sheath that's been produced by the Schwann cell. And it turns out that the Schwann cell wraps itself around and around and around the axon and makes this protective coating, this insulator that we call the myelin sheath. If we look at how that happens, we can actually go back and look at an early embryo so if we take a look at the developing nervous system in an embryo, we find that the axons don't have any myelin sheath, but next to the axons there's a Schwann cell. And then that Schwann cell starts to wrap itself around and around and around until eventually we have the axon coated with this, you can't see the wrappings here, but with this myelin sheath. The outside of the Schwann cell is still, of course, a living cell. It's wrapped its membrane around and around the axon, but the outside is a living cell. And so that living cell part of the Schwann cell that's on the outside is called the Neurilemma. And so that outside part we call the Neurilemma. Having a Schwann cell there with a Neurilemma is important because if you have a Schwann cell with a Neurilemma, you can regenerate the axon if it gets damaged. And so this diagram is trying to show that if you had a cut through the axon, the distal portion is going to die, right? But because you have these Schwann cells, the Schwann cells can go through mitosis, and they'll go through mitosis and set up a new little tube for the axon to grow back through. And so the axon can grow back through there. Years ago, I remember I still have a nice scar on my index finger. I was chopping carrots for Nathan's lunch, and I missed and made a really nice big hack into my finger. It healed up, but for the longest time, I had no sensation in that area. Now I feel it just fine. How'd that happen? Gradually, the axon grew back through that Schwann cell tube. Lots and lots of research being directed at how do you get that tube to be regrown so that we can regenerate the axons. In the peripheral nervous system, it works pretty well. In the central nervous system, not so well, right? And it turns out when we look at the central nervous system, the myelin sheaths are not produced by axons or by Schwann cells. Instead, they're produced by specialized cells that we're going to learn about next week. It's going to be a busy week next week long. But they're produced by specialized cells that we call oligodendrocytes. And there, a single oligodendrocyte can myelinate multiple axons. So in the central nervous system, the myelination doesn't occur by Schwann cells, rather it occurs by these oligodendrocytes. And it turns out the oligodendrocytes are not very good at laying down a tube for the axon to grow through. So that if you break your neck, if you will, right, and you traumatize the spinal cord, you know that the chances are not very good that you're going to get regrowth through that spinal cord. Because we don't have Schwann cells in the central nervous system, we have these oligodendrocytes. Lots and lots of research looking at how do you stimulate the regrowth of those neurons. They're making some progress. Right? One of the biggest things they've recognized is you want to decrease as much of the trauma as you can. Right? So they work very hard at trying to stop the inflammation process, but they're discovering special nerve growth factors that can stimulate regrowth of the axons. And they're actually doing some work with synthetic little tubes that they can put in to get regrowth. Okay, back to our neuron here. So if we look at that myelin sheath, we find places where the Schwann cell actually is not found. Right? So here's a Schwann cell, there's a Schwann cell. Between the Schwann cells, there are bare axon areas that don't have a myelin sheath, and those areas are called the nodes of Ranvier. You probably already know that, right? Nodes of Ranvier. Noted there. If I have to, my puns are pretty bad. All right, so nodes of Ranvier, Telodendria, synaptic knobs we did, and collateral-branches we did. All right, so we're good on the anatomy of a neuron. We need to spend a minute talking about the connective tissues associated with axons. So when you had anatomy and you got in and started dissecting your nervous system in the cat, you came to special nerves, right? You had the sciatic nerve or the ulnar nerve, whatever the nerve was. First, what we want to kind of ask here for a minute, what was it that you were actually looking at when you said, here is a nerve? And what you were actually looking at is bundles of axons, right? Thousands and thousands of axons that had been bundled up with nice connective tissues around them. And so you learned the names of those connective tissues, maybe in the nervous system, maybe you didn't, but you certainly learned these terms when you were studying the muscle system and muscles. So wrapped around each individual axon is a special coating of connective tissue that we call the endoneurium. When you were looking at muscle, you said it was the endomysium, right? You remember the endomysium that was wrapped around. So this is endoneurium. The axons then inside here are bundled in bigger bundles that we can't actually see in this diagram, but there are some larger bundles in here, and each of the larger bundle of axons is called a fasciculus, and around it is some wrapping that we call the perineurium. You called it muscle perimysium, right? So we can't see that, but it would be in here, some different bundles. And then finally, over the top of the whole nerve, right, all of those fasciculi wrapped together would be some connective tissue that we're going to call the epineurium. So when you were looking at the sciatic nerve on the cat, what you were seeing was the epineurium that was bundling up all of the thousands of axons that were inside it. Let's classify our neurons. And we can classify our neurons by structure or by function. Okay? So we can put them in categories by structure or by function. If we do it by structure, so if we're thinking of it in terms of an anatomist's point of view, they're going to say that neurons are multipolar, bipolar, or pseudo-unipolar. Multipolar, that's what we've just been looking at. So in a multipolar neuron, we find one axon and multiple dendrites. Okay? More than one dendrite. Multipolar. If we're looking at a bipolar neuron, we're going to find one axon, one dendrite. Bipolar, only two things sticking out. Okay? Bipolar. There's one more type, and we call it a pseudo-unipolar. Sometimes people will just say unipolar, but I like to say pseudo-unipolar because pseudo means false. So if we look at a unipolar neuron, really a pseudo-unipolar neuron, what we find is that in the embryo, it's just like a bipolar neuron. There is an axon and a dendrite. But as that neuron matures, the axon and dendrite grow together. So there's really only one connection. So they call it a unipolar, but it's really a pseudo-unipolar. Right? It has an axon and dendrite, but they've grown together. Since it's only one thing that's attached, they sometimes just call it unipolar. Okay. So that's by structure, but from a physiological view, we would say let's categorize them by function. And so if we were going to categorize neurons by function, we'd say, well, some of the neurons are sensory or afferent, bringing impulses toward the central nervous system. So sensory or afferent. Some of the neurons are motor or efferent. That is, they're exiting the central nervous system, carrying impulses out. And then some of the neurons would be internuncial neurons. The ones in the middle, right? The controllers, the internuncial. 99% of all your neurons are internuncial neurons.
What did you say? They're in the middle?
They're in the middle. They're between the afferent and the efferent. They're the ones that are allowing you to think, to ask those questions, right? The internuncial neurons. All right, so that was a review of the anatomy of a neuron. Old hat, right? You've been there, done that. That was straightforward.